Hello friends, welcome back to my channel Xgene and Proteinx. In this tutorial video I'll discuss about the signed Dalgarno sequence. So to know about the signed Dalgarno sequence you have to know the translation machinery. So that's why I would like to give you just a brief idea about the translation machinery. So here you see this is the codon. You see that this is the codon sequence, and this is the mRNA. This is the mRNA, and here this is the 5' region, and this is the 3' region. In between also there is one start codon, and one stop codon present. AGGAGG. So this sequence is called ORF, open reading frame. So open reading frame must contain one start codon and one stop codon. So here also the one important role plays by the ribosome. The ribosome, we know that ribosome, it has the two subunits. One is small subunit and another one is a large subunit, and in the small subunit also it contains 16S ribosomal RNA, and actually basically the ribosome also it consists of protein and the ribosomal RNA. Alright, so what happens actually normally in case of the translation, so in the ribosome it comes and it sits here in the untranslated region. It's not even the translation initiated even. So here it just binds somewhere in the untranslated region part. So this is the UTR part, untranslation region part, and also here the ribosome it gonna recognize the start codon and it bind there. That is the ribosome binding site, and the ribosome it bind there, and then the translation initiation occurs. Alright, so now the question is that how this ribosome it gonna find or recognize this ribosome binding site or start codon. So I know many of you already know this answer, but the answer is there is one consensus conserved sequence, and that sequence is responsible to help ribosome to recognize this start codon, and this sequence is known as the signed Dalgarno sequence. The signed Dalgarno sequence is present actually in the 8 nucleotide sequence apart from the start codon towards the 5' region. So what does that mean? It does that mean that that in there are 8 nucleotide sequence is apart, like 1 2 3 4 5 6 7 8, right? This is the 8 nucleotide sequence, and here the signed Dalgarno sequence is present. The sequence is AGG, AGG. So this is the signed Dalgarno sequence. So how then this kind of sequence is recognized by how they scan the ribosome then? So I already said that ribosome contain the rRNA and the protein. So rRNA it contain the complementary sequence of this signed Dalgarno sequence. Then the signed Dalgarno sequence easily bind with this region, and the ribosome it gonna find or recognize the start codon, and it bind there in the ribosome binding site, and then translation initiation occurs. So this is how it works actually. And the signed Dalgarno sequence, as I showed before, that COJAK sequence is only for eukaryote, and signed Dalgarno sequence is for specifically for the prokaryote. This is not for the eukaryote. Alright. So this is how this signed Dalgarno sequence it works. And also in the eukaryotic system, in the prokaryotic system, if like people they're gonna purify any recombinant protein by using the E. coli expression system, it should have the signed Dalgarno sequence. Then the output of the proteins or concentration of the protein will be higher in that case. So I have another question. The question is that what would happen if there is no signed Dalgarno sequence in the mRNA at all? So what would happen then? So the answer is that the ribosome still gonna recognize the ribosome binding site, but the expression of the protein, it should be very lower. The yield of the protein, it will be very low in that case. So if there is signed Dalgarno sequence, then it gonna be very easily it can detect, and the ribosome can be bind in that site, and in this case the output or yield of the protein will be very higher. Alright, so this is it guys. I hope this video will be helpful, and if you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks.